Quiz 3.1 Review. Our first question asks us to complete several mole conversions. So the first question is asking how many molecules are going to be present in 32 grams of CO2. So we're going to start with our 32 grams of CO2 over 1. And we want to get this to molecules. So we are starting with grams of CO2 and we want to get to particles. So our plan here is going to be to change grams into moles and then moles into particles. So I'm going to convert these grams of CO2 into moles of CO2. And then my next step is going to be to convert mole of CO2 into particles or molecules of CO2. So we got a couple conversion factors to fill in here. So my first one has grams and moles. This means that I need to find the molar mass of CO2. So carbon has a molar mass of around 12, and oxygen has a molar mass of 16, and there's 2 of them in CO2. So that's going to give me 12 plus 32, or 44 grams per mole. That means that 1 mole of CO2 has a mass of 44 grams. My next conversion factor is for molecules and moles. So the number of molecules in a mole is a constant. So 1 mole is always 6.02 times 10 to the 23rd things, whether those are molecules or atoms or particles. That's how many things are in 1 mole. So solving for my answer here, I'm going to get 4.38 times 10 to the 23rd molecules of CO2. So make sure whenever you do calculations that involve numbers in scientific notation that you're putting parentheses around those numbers when you put them in your calculator. The next question says how many liters of gas are present in this many grams of O2. So I'm going to start with my 15.5 grams of O2 over 1. And looking at my mole roadmap here, this time we're starting with grams and we're asked for liters. So we're going to change grams to moles and then moles to liters. That's our plan for this problem. So I'm going to go ahead and change my grams of O2 into moles of O2. And then I'm going to change my moles of O2. I need to change this into liters of O2. So my first conversion factor here is grams and moles. So once again, we need to find the molar mass of O2. So oxygen weighs 16 and there are 2 of them, which means the molar mass of oxygen is going to just be 32. So 1 mole of O2 is 32 grams. The next conversion factor is moles and liters. So if you have any gas, if you have 1 mole of any gas, it's going to take up 22.4 liters of volume. So when I solve this here, I get 10.85 liters of O2. Last question on this page is asking how many molecules of water are present in 2 liters of water vapor. So I've got 2 liters of water vapor. So this is gaseous water. And I want to convert this to molecules. So this time we're going to start with our liters and we want to get this to molecules. So we're going to change liters to moles and then moles to molecules. So I'm going to go ahead and put my liters of H2O on bottom to cancel here. And we want to convert this into moles of H2O. And then I want to convert my moles of H2O into molecules of H2O. So my first conversion factor has moles and liters. So anytime you have 1 mole of a gas, it's going to take up 22.4 liters of volume. And then my next conversion factor is molecules and moles. So 1 mole of anything is going to be 6.02 times 10 to the 23rd things. So in this case, 1 mole of water is going to be 6.02 times 10 to the 23rd water molecules. Again, we have a number in scientific notation, so make sure you use parentheses when you type that in your calculator. And the answer I get here is 5.38 times 10 to the 22nd, and that is going to be in molecules of H2O. The next question shows us some molarity calculations here. So we need to remember that molarity is equal to moles of solute over liters of solution. Now, sometimes the amount of solute is given to you in grams. Sometimes the liters of solution are actually given to you in milliliters. So we have to do some conversion factors here. Now, we notice on this chart that we have both moles and grams. So that tells me that I'm going to probably need to find the molar mass for these solutions. And all of these are NaCl solutions. So to find the molar mass of NaCl, Na has a mass of 22.99 and Cl has a mass of 35.45. So these values are just coming from our periodic table. So then when we add these two together and combine them, we get a molar mass of 58.44 grams per mole. So this means that if I have 1 mole of NaCl and I put it on the scale, it would weigh 58.44 grams. So let's go ahead and take a look at row A. So in row A, we have grams and we have liters, and we are asked for molarity and moles. So let's first take care of moles because I need moles to find molarity. So if I have 58.44 grams of NaCl, and we want to convert this from grams of NaCl to moles of NaCl, so we already found our molar mass. 1 mole is 58.44 grams. So this means that I have 1 mole of NaCl. So then to find my molarity, I'm going to need to take my moles, which we just said was 1 molar, and we're going to divide this by our liters, which is 0.5 liters. So this is going to give me a molarity of 2. 1 divided by 0.5 is going to give me 2 for my molarity. So for solution B, we have a different solution here. This time we're given the moles and the liters, and we are asked for grams and molarity. So let's handle molarity first since I already have moles and liters. So 0.15 moles divided by 1 liter is going to give me a molarity of 0.15. Now we want to figure out how many grams that is. So if I have that 0.15 moles, this time we want to change moles into grams. 1 mole of NaCl weighs 58.44 grams. So when I solve this, this is going to tell me how many grams of NaCl are in that 0.5 moles. So when I solve this, I get 8.77 grams. Our last solution, we are given the molarity and the moles, and we are asked to find the liters and the grams. So let's start with our molarity and moles to find liters. So my molarity is 0.85, and there is 0.55 moles, and we are solving for the amount of liters. So in order to solve this, I'm going to end up with 0.55 divided by 0.85. And so when I solve that ratio there, that fraction, I'm going to get that the liters, X, is going to be 0.647 liters. So about 650 milliliters here. Now to get grams, we already know the moles of my NaCl. It's 0.55 moles. So we're going to change mole of NaCl into grams of NaCl using molar mass. So when I solve for my grams of NaCl for this solution, I get 32.14 grams. The next question is asking about prokaryotic and eukaryotic cells. So we're going to determine for each of the following statements whether that is prokaryote, eukaryote, or both. So for the first one, type of cells generally considered to be more advanced, those are going to be my eukaryotes. Mitochondria are going to be found in both. So mitochondria are needed to produce energy from sugars, from food. So all types of cells need some way to do that. So eukaryote and prokaryote, they both have mitochondria. A nuclear membrane is only going to be found in eukaryotes because eukaryotes have a nucleus where DNA is stored. In prokaryotes, DNA is just found in the cytoplasm. Now, the only life form for billions of years, prokaryotes are much simpler cells, and those evolved much sooner than eukaryotic cells. And bacteria are also considered prokaryotes. Plant and animal cells are eukaryotes. That's because they have all of those membrane-bound organelles and a true nucleus. The cell membrane, ribosomes, and cell wall, those are only found in eukaryotic cells, some plant, some animal. The next question asks us to match the structure to the function within the cell, and we're going to indicate if the structure is found in prokaryotes or in eukaryotes, plant and animal cells. So my first blank is assembles proteins and lipids. So assembling proteins and lipids is going to be my endoplasmic reticulum. And the endoplasmic reticulum is not present in prokaryotes, but it is present in plant and animal cells. Shapes, supports, and protects the cell is going to be the cell wall. So the cell wall is found in some prokaryotes, and it is also found in plant cells, but not in animal cells. Synthesizes proteins is going to be the job of ribosomes. So those ribosomes are present in all cells. Converts solar energy to chemical energy in the form of sugar. So that is going to be our chloroplasts. And chloroplasts are not found in prokaryotes, and they are found in plant cells, but not in animal cells. Storage for materials are going to be our vacuoles and vesicles, so that's G. And those are not found in prokaryotes, but they are in both plant and animal cells. Contains DNA, that's going to be our nucleus. So there is no nucleus in prokaryotes, but we do have it in eukaryotic cells, both plant and animals. Regulating what enters and exits the cell is the job of the cell membrane, and all cells need to control what enters and exits the cell, so it's present in both prokaryotes and eukaryotes. Organizing cell division. So organizing cell division is the job of the centrioles. And some of our prokaryotes will have centrioles, and they will be present in eukaryotic cells. Next, we have modifies, sorts, and packages proteins. So that is the job of the Golgi apparatus. And the Golgi apparatus is not present in prokaryotes, but it is in eukaryotes. Converting our chemical energy from food into usable energy, that's going to be our mitochondria. So mitochondria are not found in prokaryotes, but they are found in eukaryotic plant and animal cells. Breaks down materials, that is the job of our lysosomes. So lysosomes are going to be present in our eukaryotic cells, but not in our prokaryotic cells. So then that leaves maintains cell shape and moves cell parts. Those are going to be the cytoskeleton. So inside of our prokaryotes, they instead have protein filaments. They don't really have a true cytoskeleton, but plant and animal eukaryotic cells do. The next question is about types of cell transport. So, how the cell can move materials in and out of the cell. So, requires a concentration gradient. This is going to be true for fusion and facilitated diffusion. Molecules move from high to low concentration. That is diffusion. If you want to move molecules in the opposite direction against the concentration gradient from low to high, the cell is going to need to put energy in to do that. So that would be active transport. Fastest when there is a large concentration gradient, that's going to be diffusion. So the bigger in the difference between the areas of concentration, the bigger the concentration difference, the faster it will diffuse. Requires the cell to use energy in the form of ATP, that would be our active transport. And using channel proteins to move molecules would be facilitated diffusion. The next question is about tonicity. So first it says to describe a situation that would cause a cell to swell. Is the solution hypertonic or hypotonic? So if the cell is going to swell, we're going to have a situation where water is entering our cell. So in order for water to enter the cell, it must be that the concentration of solutes inside the cell is much greater than outside of the cell. So this would give us a hypotonic solution, where water is going to enter the cell to try to make the concentration of those solutes the same inside and outside. The opposite situation would be one where the cell has a much lower concentration of solutes than the extracellular solution, than the solution that is outside of the cell. So I'm drawing lots and lots of solutes here to indicate that the concentration out here is much higher than inside the cell. So to try to make those concentrations equal, the water inside the cell is going to exit the cell. So this would be hypertonic. So whenever we say hypo and hypertonic and isotonic, we are referring to the solution that the cell is in, not the inside of the cell itself. So if the concentration of solutes is less inside than outside, that means water is going to exit the cell and cause it to shrink and shrivel. So this would be a hypertonic solution. Now, if we were to put a cell in an isotonic solution, then the concentration of solutes inside and outside are the same. That's what iso means. Iso means same. So this means that the cell would remain the same size because the flow of water into and out of the cell is going to be equal. Some other items that I recommend that you review are exit tickets, any additional practice sheets you were given based on your exit ticket self-score, classwork 1 to classwork 7 in the unit packet, and homework 1 to homework 4 are all going to be useful things for review on this test.